The immune system is constantly active, performing round-the-clock surveillance to protect the body from internal and external threats. Every day, as cells divide and replicate, small errors can occur in DNA due to aging, environmental toxins, radiation, infections, or normal metabolic processes. These errors can cause cells to behave abnormally, growing faster than they should, or ignoring signals that tell them to stop dividing. Such abnormal cells have the potential to become cancerous if they are not controlled early. Fortunately, the immune system has evolved specialized mechanisms to recognize and eliminate these cells long before they form a detectable tumor. This protective process is known as immune surveillance. Immune cells are trained to distinguish between healthy cells and abnormal or damaged cells. Cancer cells often produce altered proteins on their surface or display stress signals that alert immune defenders. T cells can recognize these abnormal markers and trigger targeted cell death, while natural killer cells can destroy suspicious cells without prior recognition. Macrophages and dendritic cells also play a role by engulfing abnormal cells and presenting their components to other immune cells, strengthening the response through this coordinated effort. The immune system silently removes thousands of potentially dangerous cells every day without any noticeable symptoms. Scientific evidence strongly supports this idea. People with weakened immune systems, such as organ transplant recipients or individuals with untreated HIV, have a significantly higher risk of developing certain cancers. This increase occurs because immune surveillance is impaired, allowing abnormal cells to survive and multiply. Even in healthy individuals, immune surveillance explains why cancer does not develop as frequently as one might expect, given the number of cellular mutations that occur daily. The immune system acts as an internal quality control system, constantly scanning tissues and correcting threats before they escalate. This process is not flawless. Some abnormal cells may temporarily escape detection, especially if they closely resemble normal cells or if immune responses are reduced by stress, poor sleep, aging, or chronic inflammation. However, under normal conditions, immune surveillance remains one of the most powerful natural defenses against cancer, quietly operating in the background, maintaining balance and preventing disease long before symptoms appear. A continuous biological watch that protects the body at a cellular level every moment, without rest. Cancer develops when tumor cells find ways to escape, weaken, or manipulate the immune system, allowing them to grow unchecked in a healthy body. Abnormal cells are usually detected early because they display unusual signals on their surface. However, cancer cells are highly adaptable. As they evolve, they undergo changes that help them blend in with normal cells, making immune recognition more difficult. Some tumor cells reduce or completely remove the markers that immune cells rely on to identify threats, effectively becoming invisible to immune surveillance. When immune cells cannot recognize a cell as dangerous, they do not attack it, allowing the tumor to survive and expand. Another major strategy cancer uses is immune suppression. Tumor cells can release chemical signals that weaken nearby immune cells or prevent them from functioning properly. These signals can reduce the activity of T cells and natural killer cells, which are crucial for destroying cancerous cells. In some cases, tumors attract immune cells that actually protect the cancer rather than fight it, creating an environment that supports tumor growth. This manipulation turns the body's own defense system into an unintentional ally for the disease. Cancer can also exploit natural immune control mechanisms. The immune system has built-in brakes to prevent excessive inflammation and damage to healthy tissue. Tumor cells take advantage of these brakes by activating immune checkpoint pathways, which shut down attacking immune cells. When these checkpoints are triggered, immune cells become exhausted and stop responding, even though the cancer is still present. This exhaustion allows tumors to persist for long periods without being eliminated. The tumor microenvironment plays a critical role in immune evasion. As cancer grows, it creates a surrounding environment that is low in oxygen, highly acidic, and filled with suppressive molecules. This hostile environment weakens immune cells and limits their ability to penetrate the tumor. Even when immune cells reach the tumor site, they may be unable to function effectively due to lack of nutrients or continuous suppressive signaling. Over time, this creates a protective shield around the cancer. Chronic inflammation and prolonged immune activation can further contribute to immune failure. When the immune system is constantly stimulated, immune cells may lose their effectiveness and responsiveness. This reduced efficiency allows cancer cells that initially survived immune attacks to multiply and form larger tumors. Through disguise, suppression, exhaustion, and environmental control, cancer gradually overcomes immune defenses, transforming from a few abnormal cells into a serious disease that the immune system alone can no longer control. Immunotherapy is based on the understanding that the immune system already has the ability to fight cancer, but is often prevented from doing so effectively. Unlike traditional treatments such as chemotherapy or radiation, which directly damage or kill rapidly dividing cells, immunotherapy focuses on restoring or enhancing the natural immune response. Cancer cells frequently disable immune attacks by activating inhibitory signals that keep immune cells in a suppressed or exhausted state. Immunotherapy works by interrupting these signals, allowing immune cells to regain their strength and function. Instead of targeting the tumor itself, these treatments empower the body to recognize and destroy cancer cells on its own. One of the most important advances in immunotherapy is the development of immune checkpoint inhibitors. These drugs block proteins that act as brakes on immune cells, particularly T cells. When these brakes are released, T cells can once again identify cancer cells as threats and launch a strong, sustained attack. This approach has shown remarkable success in cancers that were once considered difficult or impossible to treat. In some patients, tumors shrink dramatically or stop growing altogether because the immune system remains active long after treatment begins. Another powerful form of immunotherapy involves modifying immune cells to improve their cancer-fighting abilities. In therapies such as CAR cell treatment, a patient's own T cells are collected and genetically engineered in a laboratory to better recognize specific cancer markers. These enhanced cells are then returned to the body, where they seek out and destroy cancer cells with precision. This method demonstrates how reactivating and guiding the immune system can produce targeted and long-lasting responses without harming most healthy tissues. Cancer vaccines and immune-stimulating agents also play a role in reawakening immune defenses. These approaches train the immune system to recognize cancer, specific antigens, strengthening immune memory, and improving long-term protection. By exposing immune cells to these markers, the body becomes better prepared to respond if cancer cells reappear. This strategy shifts cancer treatment from short-term attack to long-term immune vigilance. The effectiveness of immunotherapy highlights a major shift in cancer treatment philosophy. Rather than viewing the immune system as a passive bystander, modern medicine now treats it as an active partner in therapy. By removing suppression and restoring immune balance, immunotherapy demonstrates that reactivated immune defenses can control or eliminate cancer in ways that were previously unimaginable, offering durable responses and renewed hope through the body's own natural defenses. The immune system relies on a coordinated network of specialized cells to detect and eliminate cancer, with T cells, natural killer cells, and macrophages playing central roles in this defense. T cells are among the most precise weapons of the immune system, capable of recognizing specific abnormal proteins displayed on the surface of cancer cells. Once activated, they can directly induce programmed cell death in these cells, or release signaling molecules that recruit additional immune forces. Their ability to remember past encounters also allows them to respond more efficiently if similar cancer cells appear again, providing long-term immune protection. Natural killer cells act as rapid response defenders, targeting abnormal cells without requiring prior identification. They are particularly effective against cancer cells that attempt to hide by reducing surface markers normally recognized by T cells. This makes natural killer cells essential for eliminating tumors that escape other immune mechanisms. By releasing toxic granules, they can quickly destroy suspicious cells before they multiply or spread. Their speed and lack of dependence on specific recognition make them a critical first line of defense against cancer. Macrophages serve as both attackers and coordinators within the immune system. These large immune cells engulf and digest damaged or cancerous cells, removing them from the body. In addition to this direct action, macrophages process fragments of cancer cells and present them to T cells, helping activate a stronger and more targeted immune response. They also release chemical signals that shape immune activity, influencing inflammation and tissue repair. When functioning properly, macrophages help maintain a hostile environment for tumor growth. The interaction between these immune cells is essential for effective cancer control. T cells depend on signals from macrophages and other antigen, presenting cells to become fully activated, while natural killer cells complement T cell activity by targeting cells that evade recognition together. They form a layered defense system that reduces the chances of cancer survival. However, cancer can disrupt this coordination by altering immune signals or converting macrophages into tumor, supporting cells, weakening the overall response. The balance and efficiency of these immune cells determine how well the body can resist cancer development. When immune communication is strong and activation is sustained, tumors may be eliminated early or kept under control for long periods. This complex cellular teamwork illustrates how the immune system operates as an integrated force rather than a single weapon, relying on cooperation, adaptability, and constant vigilance to defend the body against cancer. Lifestyle factors play a significant role in shaping the strength, balance, and effectiveness of the immune system, which in turn influences the body's ability to resist cancer development. The immune system is highly sensitive to daily habits because immune cells require energy, nutrients, and proper regulation to function optimally. Sleep is one of the most critical factors, as immune activity follows a circadian rhythm. During deep sleep, the body releases signaling molecules that support immune cell repair, memory formation, and coordination. Chronic sleep deprivation disrupts these processes, reducing the effectiveness of immune surveillance and increasing inflammation, a condition that can support cancer growth. Nutrition directly fuels immune defenses. Immune cells depend on vitamins, minerals, proteins, and healthy fats to divide, communicate, and perform their protective roles. Diets rich in fruits, vegetables, whole grains, and lean proteins provide antioxidants and micronutrients that protect immune cells from damage. In contrast, excessive consumption of processed foods, sugar, and unhealthy fats can weaken immune responses and promote chronic inflammation. Inflammatory environments reduce the immune system's ability to detect abnormal cells and may allow early cancerous changes to persist undetected. Physical activity also has a powerful influence on immune health. Regular moderate exercise improves blood and lymphatic circulation, allowing immune cells to move efficiently throughout the body. This increased circulation enhances immune surveillance, making it easier for immune cells to encounter and eliminate abnormal cells. Exercise also reduces stress hormones and inflammation while supporting metabolic balance, all of which contribute to a more responsive immune system. Sedentary behavior, on the other hand, is associated with immune dysfunction and higher cancer risk. Stress management is another critical factor. Chronic psychological stress triggers the release of hormones that suppress immune activity and reduce the production of protective immune cells. Prolonged stress can impair communication between immune cells, making responses slower and less coordinated. Over time, this weakened state may reduce the body's ability to control abnormal cell growth. Practices such as mindfulness, relaxation, social connection, and adequate rest help restore immune balance and resilience. Avoiding harmful exposures is equally important. Smoking, excessive alcohol consumption, and environmental toxins directly damage immune cells and DNA, increasing mutation rates and weakening immune defenses. By supporting the immune system through healthy lifestyle choices, the body becomes better equipped to detect early cellular abnormalities, regulate inflammation, and maintain internal stability, creating conditions that reduce cancer risk and strengthen long-term immune protection.